Yes, tetracycline, and the antibiotic is incorporated into tissues, mineralized at the time of the injection, and there is continuity at the edge here. You can see of the section where the original predentive recurs upward at the side of the hyaline layer. And now this is the Y-shaped structure and incorporation of tetracycline. And here, uh, the cells of the dental follicle, the cells of the dental follicle, yes, let's see this, these cells of dental follicle cells, yes, they give rise to cementoblast. Here you can see cementoblast, fibroblast of periodontal ligament, and also osteoblast of the periodontal ligament. You know that periodontal ligament contains osteoblast, cementoblast, also osteoclast, and so on. There is evidence that cells from the apical region of the dental papilla migrate around the epithelial root sheath and supplement the number of cells in dental follicle. And cells may also migrate into the developing periodontal ligament from the endosteal spaces of the adjacent alveolar bone here. During the formation of the root and periodontal ligament, there is obviously dramatic increase in the number of connective tissue forming cells such as cementoblasts, fibroblasts, osteoblasts, and the stem cells exist within the region to allow for this, perhaps lying in the perivascular location. Here you can see that this periodontal ligament is highly vascularized, and these stem cells they surround these blood vessels, and they have such perivascular location. But it is not clear whether the three main types, these cementoblasts, fibroblasts, and osteoblasts, have arise from a common progenitor cell or stem cell, or whether each type has its own specific stem cell. And yes, to the main connective tissue forming cells, osteoblasts, osteoblasts, cementoblasts, and fibroblasts. There are also osteoclasts. If you remember from periodontal, we discussed this in periodontal ligament cell types. There are also osteoclasts. Osteoclasts, you know that they are resorbing cells, osteoclasts, and the osteoclasts they are derived in bone marrow, and their progenitor cell is a monocyte, and this monocyte give rise to macrophages, to dendritic cells, to microglia, and to osteoclasts. As tooth erupts, there is little evidence of any significant bone deposition at the base of the sockets, but resorption often being prominent at this site. At these sites in sockets, tooth sockets, and there are a bone deposition at these sites is precluded as a cause of tooth eruption, and here you can see that. Let's see here, this is diaphragm, and proliferation of the cells of the epithelial diaphragm is accompanied by proliferation of the cells of connective tissues of the pulp adjacent to the diaphragm. This induces the formation of and differentiation of these mesenchymal or papillary cells, and they, differentiate into the odontoblasts, and the free end of this diaphragm does not grow into the connective tissue, but the epithelium proliferates coronal to the epithelium diaphragm. And you know that this diaphragm, differential growth of epithelial diaphragm here in the multi-rooted teeth, causes the division of root into two or three roots, Yes, and before division of the root trunk occurs, free ends of the horizontal epithelial flaps grow towards each other and form such and separates the common canal. Yes, it is one rooted, single rooted, it is two rooted and three rooted canals. Now, mm, yes, and this, you know that after disintegration of epithelial root sheath, this is periodontal ligament, this is the cementum, and this is the cementa periodontal junction. And here you can see the remnants of this epithelial root sheath, and these are epithelial rests of molasses. And you can see that the distribution of the epithelial rest cells on the root surface, and this rest form network in the periodontal ligament that parallels the root surface. Yes, now let's see these structures. These are the erupting permanent molar, or and this is just emerging into the oral cavity, and you can see that there is the periodontal developed periodontal ligament, and also you can see the fibers in periodontal ligament. And here it is erupting second premolar, and you can see this is erupted first premolar, and there is absence of periodontal ligament fibers here. And let's now discuss cementogenesis. You know that periodontal ligament, yes, periodontal ligament consists of cementum, alveolar bone, and periodontal ligament, and the gingiva, which is the soft tissue, which is the part of oral mucosa and part of the periodontium. And we will not discuss this gingiva, only hard part and the cementogenesis from this periodontium. Now about cementogenesis, here, what is it? It is the showing mineralization. No, cementogenesis. Uh, and yes, cementogenesis will be considered in the terms of formation of acellular cementum and cellular cementum. There are four or five types of cementum, different types of cementum, uh, which are distinguished by fiber content, cells, and position. And you can see this here. Classification on cementum. This classification is based, for example, to on development. For example, yes, primary cementum and secondary cementum, according to the development. Yes, according to the its cellularity, there is acellular cementum and is cellular cementum. According to the origin of the collagenous matrix, such as extrinsic fiber cementum, then intrinsic fiber cementum, there is a mixed fiber cementum, and is a fibrillar cementum. Yes, and Schroeder's classification, there are only acellular. According to authors, there are some different classifications. There is acellular, afibrillar cementum, acellular extrinsic fiber cementum, and cellular mixed stratified cementum. And you can see the cementum location. Cementum is, with about recall cementum, you know that cementum is one of the mineralized connective tissue. It is not vascularized or innervated, and it covers the outer surface of the tooth root in a thin layer. The fibers of periodontal ligament insert into the cementum. In the cementum, these fibers are known as Sharpie fibers. You know already, we discussed it with cementum. And the periodontal ligament fibers attach to the teeth to their bony sockets. This is why cementum is also part of the periodontium. Cementum first was described in 1835 by pupils of Parkingi in Prague. A distinction or is now made between five different types, and we will discuss these five different types of cementum in humans. And these types differ with regard of structure, fiber composition, and cell content, but also in terms of localization. And let's discuss the Yes, here you can see acellular cementum, cellular cementum. Yes, acellular cementum is first formed, cellular secondary formed, secondary, present on the cervical third of half of root. And here you can see these parts. This is acellular, afibrillar cementum. Yes, and here you can see a cellular but extrinsic fiber cementum when the fibers from periodontal ligament penetrate the cementum. Yes, here this is from outside. They have no cementocytes, and from extrinsic, from outer, externally embedded into the cementum. These part are extrinsic, acellular extrinsic fiber cementum. And here you can see this blue appearance. This is cellular intrinsic fiber cementum and cellular mixed fiber cementum. And the first of all, let's discuss the formation of cementum. Because it is very closely associated with the development of the tooth root. After formation of the crown, after crown formation, and when this process is complete, the inner and outer epithelial face each other. The nomenclature of the epithelium is initially known, as the cervical lobe. From it, the Hedwig epithelial root sheath emerges, which defines the shape during root formation, and this involves continued cell division in the apical region while the coronal portion disintegrates. The cells of the Hedwig epithelial root sheath form a circumferential surface onto which the first deposits the root dentin. It is necessary for cementum deposition. First, the deposits the dentin. Yes, yes, and in this way, it shapes the root. Do you see this algorithm of this process? First of all, there is the sheath of Hertwig double layered sheath, and then root sheath. Then when this odontoblast, it causes the differentiation of odontoblast then secrete dentin. After this Hertwig's root sheath disintegrates, and only after this cementoblast appear and differentiate, and they form this they begin the deposition of cementum. When advancing root formation and Hertwig epithelial root sheath migrates slightly further in the apical direction, and is coronally, it continues to disintegrate, but not entirely. A loosely woven mesh of epithelial rests, you know, that persists, and this continues to disintegrate. These parts are these remnants are these epithelial rests of molasses. Yes, rest of the region and around the root apex, which is just forming, is rich in ectomesenchymal progenitor cells. You know that this apical part, it consists of these progenitor cells or stem cells, and from which these fibroblasts, preodontoblasts, precementoblasts are derived. These stem cells give rise to all these cells. These cells are periodontal ligament cells. The epithelial cells from the heteric epithelial root sheets can act on the progenitor cells, making use of the close proximity, and thus preodontoblasts, among other cells, originate from the progenitor cells, and they secrete polypeptides that are similar to enamel and amelogenin. This process has not been detected for cementoblasts. In cementogenesis, the Hartwig epithelial root sheath disintegrates enough to enable the dentin surface of the freshly formed root to come into contact with cells of dentinal follicles. Only after is thought that after this the differentiation of connective tissue cells into cementoblasts takes place. Deposition of cementum onto this dentin surface starts as soon as freshly formed surface of root dentin are freed of their covering with Hertwig's epithelial root sheet. Yes, and after this cementoblasts begin formation of cementum. Uh, Now let's discuss. You know that there are cementoblasts, cementocytes. Cementocytes are already embedded in the organic matrix cells, and cementoblasts are effector cells. They formative cells. They form cementum. And now let's discuss the formation of acellular afibrillar cementum. Here are the types of acellular cementum. Acellular cementum is first secreted cementum, and here you can see it is it has such glassy appearance without cells. Here you can see cellular cementum, and there is also intermediate cementum, but it is now thought that is the artifact is intermediate cementum. And also you can see a cellular cementum here. You know that sometimes this cementum at cemento enamel junction, this cementum covers overlaps the enamel, yes, and then this disintegrates, and this forms such a fibrillar cellular cementum. And this type of cementum, acellular afibrillar cementum, contains no cementocytes and no collagen fibrils. It is the first type of cementum to be formed during tooth development. It covers parts of cervical enamel and rarely the cervical root surface. When the epithelial covering of the freshly formed coronal enamel disappears, the adjacent connective tissue comes into contact with the enamel surface. Here you can see pre-eruptive maturation of the enamel is completed at this stage, and this occurs shortly before eruption or as the tooth is just starting to erupt. At this stage, or even after eruption, cementoblasts deposit initial layers of a cellular afibrillar cementum here, which is homogeneously mineralized. As this is acellular cementum, it is assumed that serum proteins and minerals precipitate together on the enamel surface without the involvement of cementoblasts. This type of cementum was formerly also known as coronal cementum. It has long been known to exist in herbivores whose dental crowns are regularly covered with cementum, even in the enamel organ. In humans, this type of cementum is ultrastructurally distinguishable from the other types of cementum. And it cannot be distinguished from acellular fibrillar cementum. This type of cementum is also present as cemental spurs and cementum pearls. You know cementum pearls, and we have the picture of cementum pearls on the dental enamel. And cemental spurs originate at the cervical margin of the enamel and overlap the cervical enamel margin to a variable extent. Now, here, what is it? Here you can see acellular. This was acellular, yes, acellular, afibrillar cementum. And let's recap. It consists of mineralized matrix. Here you can see at the margin, at the gingival margin and gingival epithelium. You can see at sulcus, epithelial sulcus, and it contains neither cells nor extrinsic or intrinsic collagen fibers. This cementum does not have any function in tooth attachment, and it is less homogeneous, and was described by Schroeder in 1986. And these connective tissue cells are responsible for acellular afibrillar formation when they come in contact with enamel surface, and it is a product of cementoblast and is found in coronal cementum. Let's now discuss the cellular extrinsic fibers. You know that these extrinsic fibers are come from periodontal ligament. You know that periodontal ligament, it is between the tooth root and alveolar bone, yes. And the fibers from periodontal ligament, they propagate into the cementum, freshly made the cementum, and into the bone, yes. And it attach this cementum or tooth to the alveolar bone. And this acellular extrinsic fiber cementum originates pre and post eruption, and as a result of the deposition of cementum matrix, most of the cementoblasts are not enclosed but shifted to the surface. And let's see here or here. Let's see here. This is acellular. Firstly is deposited acellular cementum and then cellular cementum. And here you can see acellular cementum layer, and after this cellular cementum layer. And this is a thin layer of acellular cementum. Yes, from most of the cementoblasts, yes, because they move on the surface. Therefore, this type of cementum is predominantly acellular. The extrinsic fibers are collagen fibers that radiate from the periodontal space into the layer of cementum. They are called, Sharpie fibers, and anchor the tooth in the bone. Yes, this type of cementum covers 60 to 90% of the root length as a deposition directly into the freshly formed dentin matrix of the root. Thus, this cervical third of all the teeth almost exclusively contains acellular cementum, extrinsic cementum. In this apical region, a cellular extrinsic fiber cementum only develops as a second layer, because the first layer in this region consists of primary formed cellular intrinsic layer. The formation of acellular extrinsic fiber cementum accompanies root development. In the area within 300 micrometers in the apically advancing root edge, the zone of cementogenesis shifts more apically when the Hartwig's epithelial root sheath disintegrates and makes the freshly formed dentin matrix accessible to the surrounding periodontal cells. And you can see that they are these fibers, fringe of short. This is the fringe of short collagen fibers collagenous fibrils is deposited onto the not yet mineralized dentin matrix, these fibers, and also you can see these fibers, periodontal ligament, this is dentin, and this is cementoblasts. And yes, these cells that bring these about are cementoblasts, which cannot be distinguished morphologically from fibroblasts, and these short collagen fibrils lie perpendicular to the root, perpendicular to the root axis, root surface, and their ends extending into the still non-calcifying dentin matrix. Later, when they grow longer and anchor the tooth into the bone, they become periodontal ligament fibers, periodontal ligament fibers that traverse the periodontal space. This short fringe of short collagen fibers becomes denser coronally, and within 100 micrometers of the advancing root edge, fibroblast type cells, they make contact with the dentin surface and start to assemble the fibril fringes into bundles that correspond to fibers. Yes, and these fibers, this fringe of fibers becomes even denser, and together with the cells that create it, form dense cell fiber complex. Mineralization of the root dentin walls takes place in the fibers, then the mineralization front passes into the cementum layers, where the fibers are further enclosed. You know that after mineralization, these fibers are enclosed, embedded into mineralized matrix, and about 1 millimeter coronally of the growing root edge, the acellular extrinsic cementum is only a few micrometer thick. And the classification of acellular extrinsic fiber cementum is similar to that in a dentin, and stems from individual globules that fuse together and they get bigger. Further calcification happens later and among the smooth surface. Yes, and this type of cementum appears transparent and structureless in non-decalcified ground sections viewed under the light microscope, and the fiber structure only becomes visible when a ground section is etching, when a fracture specimen is viewed under scanning electron microscope. And here you can see acellular extrinsic fibers here, or cementum originates pre and post eruption as a result of the deposition of cementum matrix. Most of the cementoblasts are not enclosed but shifted to the surface, and therefore this type of cementum is acellular. And the Sharpie fibers in cementum are homogeneously mineralized, and the majority of the crystals running with their central axis parallel to the path of the fibrils, and after disintegration of the organic components, the entry points of the Sharpie fibers are visible in specimens, and the cementum surface. And let's see, this is acellular, then cellular, and this is dentina enamel junction, and first layer, which is deposited, is acellular cementum adjacent to the dentin, and it is covered by a thicker layer of cellular cementum, which contains cementocytes, lacunae, and a thin layer of acellular cementum you can see here. And let's recap cellular intrinsic fiber cementum. Here you can see that cementocytes are already embedded into the organic matrix, and they have such radiating canals which are called canaliculi, and via these canaliculi they communicate to each other. You can see here canaliculi, and this is the cementosite site in the lacuna. Embedded into lacuna, you can see the nucleus of these cells and the cytoplasm of these cells, and this is demineralized cellular cementum of a mouse molar, and shows cementocytes in lacuna surrounded by the collagenous cementum matrix. And let's now discuss the cellular cementum. And here you know that they are secondary. This is called secondary cementum. After half the root is formed, a rapidly forming, less mineralized variety of cementum is laid down, and this is cellular intrinsic fiber cementum, CIFC. And it is found in the apical third and confined to the apical and interradicular regions of the tooth. And you can see here that this is cellular cementum. And cellular intrinsic fiber cementum originates pre-eruptive in the area of root furcation and post-eruptively in the apical regions. And it is always the first layer of cellular mixed fiber cementum. And in subsequent layering of cellular mixed fiber cementum, it forms the intermediate layer. And the cellular intrinsic fiber cementum is produced solely by cementoblasts. And these are already degenerating cemento layer of cementocyte. And here you can see viable superficial cementoblasts. It contains no Sharpie fibers. These cells are distributed with a variable density, cellular intrinsic fiber cementum, and in place no cells are visible. As in a cellular extrinsic fiber cementum, the formation of cellular intrinsic fiber cementum starts with the deposition of cementum directly onto the fresh dentin layer. Dentin layer is necessary for cementoblast functioning. Keep in mind that dentin layer gives signals for cementoblast differentiation and deposition of cementum. When the Hartwig epithelial root sheath disintegrates and thereby leaves the root surface clear, a relatively large amount of cementum matrix is rapidly formed along a zone from 100 to 200 micrometer coronally of the growing root edge, and there are interweaving of matrix arises between the cementum and dentin at the future cemento enamel junction. Many haphazardly arranged collagen fibrils are contained in the cementum matrix, as are bundle collagen fibrils, which are arranged parallel to the root surface in this type of cementum. And the cementocytes, here you can see the cementocytes define this arrangement by creating compartments running parallel to the root surface in their cytoplasmic processes. This matrix is mineralized along mineralization front, which is roughly 200 micrometer away from the adjacent root edge, and initial mineralization takes place relatively quickly. Cellular mixed fiber cementum originates in this way, comprising layers of acellular and cellular cementum. Yes, and intrinsic fiber cementum and acellular extrinsic fiber cementum. Cellular and acellular intrinsic fiber cementum are also formed in resorption lacunae and in response to traumatic tooth damage. Here you can see the maturation occur layer by layer for the collagen fibrils, and you can see the cementum. These are cementoblasts, differentiated cementoblasts, and here you can see this mineralization front, and firstly formed the cementoid layer, which is very alike of predentin. This is outermost layer, roughly 3 to 8 micrometer wide layer, which overlies the acellular extrinsic fiber cementum in places, and it appears structureless under the light microscope. You can see here the structureless. It is eosinophilic. At its surface lie many polygonal fibroblasts, between which the periodontal ligament fibers radiate into the cementoid. In the cementoid surface is cleared of organic material, and these fiber entry points can become seen. And here you can see this micrograph, and this is developing of cementum and periodontal ligament, and it is unerupted human premolar teeth, and here you can see this fiber fringe, it is, protrudes from the thin layer of mineralized acellular cementum, and this is periodontal ligament, and this is acellular cementum. And here you can see 15 micrometer thick layer of mineralized cementum, which covers the root surface. Here you can see. And also you can see some of the periodontal ligament fibers are continuous with the fiber fringe. These short fibers are called fiber fringe. Yes, here you can see the firstly laid down acellular cementum, and here you can see the fiber fringe arrowhead are indicated by arrowhead. And um, yes, as root formation is a continuous process extending over several years, the phase on initial deposition of cementum onto the growing root also lasts 4 years. This results in a variable distribution of the different types of cementum of the root. And here you can see that the calcified root surface, which for acellular extrinsic cementum. And here you can see this is dentin, this is cementum, and you also can see these fibers, which are Sharpie fibers, and Sharpie fibers penetrate cementum, and also you can see the cells of periodontal ligament, which form, these are fibroblastic cells, they are spindle-shaped cells, fusiform cells, and they produce such collagen fibers, which assemble then and form the Sharpie fibers, and Sharpie fibers enter here. You can see the Sharpie fibers, which enter this cementum, and there further will be embedded into the cementum. To begin with, acellular extrinsic fiber cementum is formed at the neck of the tooth, and later, when root formation has advanced, more of the cellular intrinsic fiber cementum is formed. Firstly, are formed this acellular fiber cementum, and these are Sharpie fibers, and they then embedded into the cementum. Then intrinsic cementum. After the cellular intrinsic cementum is formed further apically or in the area of bifurcation of root. And this type of cementum can also spread in the coronal direction and be deposited over the acellular extrinsic fiber cementum, and conversely, further acellular extrinsic fiber cementum can be laid down over the cellular intrinsic fiber cementum, and this is called the cellular mixed fiber cementum. Cellular mixed fiber cementum, excuse me, fiber cementum, and is not really a separate type of cementum. Here you can see this is the decalcified root surface, and it forms the acellular extrinsic fiber cementum. These are the Sharpie fibers, and these cells are are cementoblasts and Sharpie fibers which penetrate between these two cementoblasts. And here you can see the cementoblast, and also you can see associated with the formation of cellular cementum. And this cell is typically more irregular, you can see, in morphology, and contains larger amount of intracellular components, organelles associated with the protein synthesis and secretion. And also you can see the precementum on cementoid, which is newly formed cementum and unmineralized cementum. And here it is developing root showing fibers, fiber fringe. Here you can see fiber fringe running parallel to the root surface with little attachment to the cementum fibers. And now here you can see Sharpie fibers. It is light microscope human periodontal ligament, and you can see the fibers with Sharpie fibers, which are embedded in the alveolar bone and also into the cementum. And here you can see this is the electron micrograph of periodontal ligament fibers inserting into the cementum, Sharpie fibers embedding into the cementum, and it is higher magnification that individual banded collagen fibrils of Sharpie fibers can be seen in the cementum. You can see these are all collagen fibers. This type of this cellular mixed fiber cementum, it is not really separate type of cementum, but it is a mixed product of alternating layers of cellular and acellular intrinsic fiber cementum, and it is regularly found stratified in certain region of the root. There may be justification of referring by the distinct name. Here you can see this is the decalcified root surface, which shows the cellular cementum formation. And here you can see the cementoblasts, these cells, cementoblast, and here you see the cementum, which is structureless part and not yet mineralized part. And here you can see two cells which are cementocytes, which becoming incorporated, embedded into the cementum matrix. And this is high magnification of electron micrograph. You can see the dentin, and you can see these extrinsic fibers or Sharpie fibers, and this is cementoblast. And you can see the Sharpie fibers which are perpendicular to the central axis of the root surface. And intrinsic fiber cementum is always laid down in the apical region as the first layer. Onto this, a thin layer of extrinsic fiber cementum is deposited, although it is not known when. This thin layer often merges into the thicker layer of pure acellular extrinsic cementum as it extensively covers the root further coronally. In the apical region, however, the thin layer of extrinsic fiber cementum is covered by one or more often thread-like layers of thin intrinsic fiber cementum, which alternate it with the thin layers of extrinsic fiber cementum. Then, very often, in addition to these four differently structured types of cementum, intermediate cementum has been described as separate fifth type, but it is supposed that to be found as a separate layer between root dentin and the cellular mixed fiber cementum. But the most recent studies have shown that these irregular and non-homogeneously mineralized intrinsic fiber cementum intermediate layer are not separated. I can show you this where it was intermediate layer. Just a moment. Here, here. No, intermediate layer. It is not a certain layer. It is the layer are not separate, but represent the boundary between dentin and cementum, and it is not a different layer. And there is also um, very pronounced in some individuals anomalous thickening of the cellular mixed fiber cementum, and the causes and condition of these are largely unknown, and such extreme cemental thickening is known as hypercementosis, cementum hyperplasia. It can be localized or occur extensively over the entire root cementum, and here you can see these are cementings, which are small mineralized bodies, which may be found in the periodontal ligament or attached to the cementum, or alveolar bone, or occur free in the periodontal ligament, and they are also called semantics. And here you can see semantic with each attached to the cemental surface within the periodontal ligament, and also you can see this yes, this picture, which is the cementum's resorption. It undergoes cementum very often undergoes resorption and repair, and the resorption of cementum by odontoclasts are osteoclast like cells, which are formed for resorption. They are similar to osteoclast, and this may occur in a variety of conditions, including orthodontic, tooth movement, and resorbed areas. You can see, and you can see resorbed areas may be filled in later with a newly deposited cementum, and these arrowheads indicate the dentin cementum junction. Yes, and let's recap this cementogenesis and cementum. The root dentin is entirely covered with cementum. Cementum is a tissue that is produced by cementoblasts. The supporting fibers of the periodontal ligament radiate into the cementum. There are different types of cementum distinguished by fiber content, cell composition, and position. These are acellular, afibrillar cementum, acellular extrinsic fiber cementum, cellular and acellular intrinsic fiber cementum, cellular mixed fiber cementum, cellular mixed fiber cementum, intermediate cementum, maybe, and cementoid. Cementoid, that is freshly made cementum without mineralization. You know that cementum is mineralized connective tissue, and it is not vascularized or innervated, and it covers the outer surface of the tooth root and is at in there. And the cementum resembles bone more than dentin and enamel. Cementum is produced and laid down throughout life, and incremental lines are identifiable under light microscope, and small isolated resorption lacunae in the cementum can be repaired by being filled up with cementum. Yes, now let's discuss the periodontal ligament. Periodontal ligament, you know that periodontium, uh, what is this? Yes, periodontium is the functional unit, and it consists from cementum, alveolar bone, periodontal ligament, and gingiva. But gingiva denotes a specific region of oral cavity. This is both oral mucosa and part of periodontium, and we'll not discuss gingiva. And we already discussed cementogenesis and cementum. And let's now discuss periodontal ligament, which is very important in tooth in supporting tooth in its sockets. You know that the alveolar process, bone, cementum, and structures of periodontal ligament, these all structures develop from the cells of the dental follicle proper. You know that tooth germ consists of three structures: enamel organ, which gives rise to enamel, then dental papilla gives rise to pulp, yes, and also dental follicle or dental sac, which gives rise to periodontal ligament, to cementum, and to alveolar bone. Let's discuss develop periodontal ligament. Yes, the portion of inner cells of the dental follicle that are involved in the development of periodontium. These are cells of dental follicle. As early as the Bell stage of odontogenesis, the dental follicle is clearly distinguishable from the other connective tissue because of its fibrous and cell-rich structure around the tooth germ. At this stage, the fibroblasts have already been characterized as highly active cells. The collagen fibers, the fibroblasts, and collagen fibers are already present here. Initially run largely parallel to the dental follicle and clearly still have no contact with the trabeculae of the young bony alveolus. As soon as tooth root is formed, the fibroblasts and the first collagen fibers line up at the angle to the cemento-enamel junction, and they run in the coronal direction along the dental crown and represent the eventual dentinogingival fibers. And the longer the tooth root becomes, the more fibers are formed and traverse in an obliquely coronal direction to the bony crypts, and which are in the process of developing. And here you can see the synonyms of periodontal ligament. This is periodontal membrane, alveolar dental ligament, desmodont, pericementum, and dental periosteum and gomphosis. These are all synonyms of periodontal ligament. You can see this periodontal ligament attached the alveolar bone to the cementum. From one side here from right side there is the cementum. From left side is alveolar bone, and this is the tooth socket. And alveolar bone proper is called this socket, is which the bone which lines this socket in which tooth is attached. Let's now see the formation of yes, the periodontal ligament develops from the dental follicle. Cells of the dental follicle that differentiate are fibroblasts, and they synthesize and form the fibers, secrete the collagen fibers, and they assemble afterwards and form these Sharpei fibers and principal fibers. And you can see the organization of periodontal ligament fibers, and this orientation of these fibers changes according to the stage of tooth eruption. And here you can see the development of these fibers. Generally, the sequence of fiber formation is the same in primary accessional and successional teeth. First, thin, short collagen fiber bundles are embedded in the freshly developing cementum. This is cementum, this is alveolar bone. Initially, they project only slightly beyond the still non-calcified cementum surface. On the bone side, from this side, only a few collagen fibers are initially embedded, which in places protrude out of the bone. Only a few collagen fibrils are seen here, and the periodontal ligament between the cementum and the bone surface is filled with the freely passing collagen fibers that run parallel to the tooth axis. the number and thickness of the collagen fibrils emanating from the bone increases. Here you can see the next stage when the fibers from alveolar bone, they increases during the subsequent development. They extend into the quarter of the periodontal ligament space close to the bone and bear a large number of ramification at their branching at their ends. At this stage, the fibers on the cementum side are still unchanged. Here you can see, but later they increase in size and branch many times. Here, branch many times toward the middle of the periodontal space, where they connect with the bone side fibers. It is not until the tooth enters into the occlusal function that the network of fibers get thicker and connects the cementum surface to the inner surface of the bony alveolus. During formation of mesh of this mesh of periodontal ligament, fibers, the collagen fibers in an area in the middle of the periodontal ligament space appear to be loosely arranged or intertwined in a lattice-like formation instead of displaying a strictly functional orientation. This area has been referred to as the intermediate plexus. Here, intermediate plexus. It was assumed that the fibers in this zone continually detach from one another and join together anew whenever the tooth alters its position. However, the intermediate plexus is no longer present in erupted teeth in full occlusal function. Here you can see this is the development of Sharpie fibers. You can see this previous picture, how these fibers emanate from the alveolar bone and here from cementum, and they then connect in the middle of this periodontal ligament and form the intermediate plexus. And the Sharpie fibers in the extrinsic fiber cementum arrange themselves differently in consecutive layers when the tooth changes its position. And here you can see these Sharpie fibers, and here you can see these Sharpie fibers, which are this is periodontal ligament, bone from one side and cementum from other side, and you can see these Sharpie fibers, these lighter lines which propagate the both bone and cementum. And here you can see that the principal collagen fibers, which are associated with the erupting permanent canine, and it is not so prominent as here. This is well differentiated collagen fibers for fully erupted permanent canine of the ferret. And this is the no. Here you can see this the development of fiber bundles of the periodontal ligament, and they are thin and not well oriented. After this, they have such orientation, and they are oriented obliquely, and only after the tooth come into occlusion, they they have such bundles of periodontal ligament, and it becomes well organized in terms of thickness and orientation. And this you know that here let's recap of the structure periodontal ligament. The periodontal ligament is a tough connective tissue structure that is rich in fibers, cells, and vessels, nerves, and ground substance. You know that other ligaments in the body, they are avascular, but periodontal ligament is very profusely vascularized. It has also nerves and the nerve endings and ground substance. The periodontal ligament attach the tooth to the alveolar bone, which is via the cementum. The resulting joint-like connection is a syndesmosis, and the collagen fiber bundles insert into the cementum and highly branched and intertwined along part of their path extend to the inner cortical plate of the alveolar bone. At the alveolar crest, the periodontal ligament merges into the connective tissue of the attached gingiva. At the apical foramen, the periodontal ligament tissue merges into the pulp tissue. The periodontal ligament fibers are formed by fibroblasts. You know that the cells of main cell population in periodontal ligament are fibroblasts, which are connective tissue cells, and they make up roughly 50% of the cells in the periodontium. And these cells form collagen fibers, and these fibroblasts of periodontal ligament originate from the dental follicle, which has formed from the cells of the cranial neural crests. Periodontal ligament fibroblasts are of ectodermal origin. By contrast, the gingival fibroblasts are derived from the local mesenchyme, and they differ in properties. Among other characteristics, periodontal ligament fibroblasts have a higher proliferation rate and form more alkaline phosphatase and cyclic adenosine monophosphate than gingival fibroblasts. And there are also other differences between the normal fibroblasts and periodontal ligament fibroblasts. And there are the periodontal ligament also, that it gives rise to the cemento progenitor cells that lie in the parts of periodontal ligaments close to the cementum. They originate from stem cells close to vessels and cannot be distinguished morphologically from inactive fibroblasts, and they become cementoblasts that migrate toward the cementum surface. The cementoblasts are rather cuboid, not as elongated as fibroblasts, and they are characterized by plasma, cytoplasm, and corresponding abundance of intracellular organelles for protein synthesis, such as endoplasmic reticulum, Golgi complexes, ribosomes, and it is they are secreting cells, and when they are secreting, the membrane displays numerous cytoplasmic processes. The nucleus is irregularly folded, and the cytoplasm is basophilic, and cementoblasts are derived from the cells of the dental follicle at the start of cementogenesis. And fiber development differs among primary, accessional, and successional teeth. And also, these osteoprogenitor cells, they also lie in the parts of the periodontal ligament close to bone, and they give rise, they are originate from stem cells close to vessels and cannot be distinguished morphologically from inactive fibroblasts. They turn into osteoblasts, and osteoblasts lie in the part of the periodontal ligament close to bone, and they are cuboidal like cementoblasts and exhibit a pale basophilic cytoplasm with extensive rough endoplasmic reticulum and Golgi apparatus. Their nucleus is round and prominent. And during the active phases of bone growth, the osteoblasts form a layer of cells that almost completely covers the freshly formed bone surface. This freshly formed bone is called osteoid, and it is yet non-mineralized part. And also, that periodontal ligament contains osteoclast, which develop from hematopoietic stem cells in bone marrow, and their shape is amoeboid and amorphous, and their polynuclear character makes them clearly identifiable. and they contain numerous mitochondria and prominent Golgi complexes, but minimal rough endoplasmic reticulum. And like osteoclasts, they are resorbing cells. They are only present in large quantities during active phases of bone remodeling. They mainly reside in depressions in the bone surface known as house shaped lacunae. And you know that also periodontal ligament contains mesenchymal undifferentiated cells, which are progenitor cells, and they are mainly found in perivascular. And they are also epithelial cells in the periodontal ligament, which are present of epithelial cells. These epithelial rests of molasses. They are remnants of the Hartwig epithelial root sheet, which, as a closed epithelial layer, provides a template into the which dentin of the root is deposited from the inside. And the disintegration of Hartwig's epithelial root sheet is the precondition for deposition of cementum on the root surface. And it seems to be normal for the Hartwig's epithelial root sheet not to disintegrate fully because there are remnants, and they give rise to epithelial rests and root pearls. Yes, and also they can form this two-dimensional network that lies close to the cementum in periodontal ligament. And you know that these collagen fibrils are arranged in periodontal ligament, and there are some fiber groups such as alveolar crest group fiber bundles, horizontal group fiber bundles, oblique groove fiber bundles, and apical groove fiber bundles. Also, they may be in multi-rooted teeth. You can find intraradicular groove fiber bundles. Then we can discuss the alveolar bone development. Alveolar bone develops from the dental follicle, and it has the ectomesenchymal development, alveolar bone, and you know that dental follicle is dental follicle contains these ectomesenchymal cells, and these ectomesenchymal cells differentiate into osteoblast, into osteoblast, odontoblast, and give rise, lay down the matrix called osteoid. Uh, and there are some ways of ossification, that mandible and maxilla develop intramembranosely. There are some ways of ossification, and these are intramembranous ossification and endochondrial ossification. And intramembranously are ossified the whole mandible and the maxilla, and in a fibrocellular condensation. Here you can see how this takes place. This is ectomesenchyme, that and some condensation takes place. And this is fibrocellular condensation. A center of ossification appears in which osteoblasts lay down first form embryonic or woven bone. As the teeth develop, bone extends from the developing mandible and the maxilla to surround and protect the teeth, forming the alveolus. And you can see the areas of mesenchyme here and here and here, and condensed mesenchyme here and here, condensed mesenchyme. They are adjacent to layers of new osteoblasts. These cells which lie, these osteoblasts lie on the surface of this osteoid, and some osteoblasts have secreted mesenchymal fibers matrices of bone, here you can see, and the surface of which remain covered by osteoblasts. These are secreted cells, formative cells, these osteoblasts, and they form such epithelial lining on this osteoid. And between the two regions of new woven bone are areas with small blood vessels, a lot of blood vessels you can see, and this is newly formed osteoid and the layer of osteoblasts. And the alveolus is separated from the developing enamel organ by the dental follicle to accommodate the growing tooth germ. The lamellae of the developing alveolar bone undergo resorption on the inner wall of the alveolus. And then here you can see this is alveolar bone, alveolar cortical plate, and alveolar bone proper. This is the lingual cortical plate and labial cortical plate. And this to accommodate the growing tooth germs, the lamellae of the developing alveolar bone undergo resorption on the inner wall of the developing inner wall of the alveolus, and there, after this, bone is deposited. Here you can see by osteoblasts lining on the osteoid, on the surface of this osteoid. Mhm. And the developing teeth therefore come to lie in a true of bone. Later, the teeth become separated from each other by the development of interdental septa. With the onset of root formation, interradicular bone develops in multi-rooted teeth. As in other sides, the collagen fibers in the newly formed alveolar bone have a more variable diameter and lack a preferential orientation, giving the bone appearance, matted appearance, lack a preferential orientation, and this immature bone turned woven bone, this is embryonic bone, it is a gel-like bone. This woven bone has a larger and more numerous osteocytes than adult bone. It is formed more rapidly and has a higher turnover rate. And here you can see that the whole body of mandible, except the anterior part, is undergoes intramembranous ossification. Ramus of mandible, as far as mandibular foramen, also undergoes intramembranous ossification, and endochondrial ossification undergoes anterior portion of the mandible symphysis, the junction of two bones, two mandibular bones, and we discussing on a next lecture with orofacial differentiation and part of ramus about the mandibular foramen and coronoid processes and condylar processes. They undergo the endochondrial ossification, and here you can see this is the intramembranous ossification, when there is mesenchymal condensation and blood. Here you can see blood vessel, and there appear ossification centers, and after these centers fuse to form such trabeculae, and there are the clefts between these trabeculae, and blood vessels grow in between. And here you can see this is the micrograph of endochondrial ossification, in which pre-existing matrix of hyaline cartilage is eroded and invaded by osteoblasts, which then begin osteoid production. And here you can see the enamel pearl, and you now know now that these pearls are developed from these are spherical masses and develop ectopically in the areas of root furcation. And these are small isolated spheres of enamel that are occasionally found on the root surface, especially towards the cervical margin. They are particularly common at the root furcation area, and they may also occur at lower levels. And And here you can see the hypophosphatasia is a condition in which there is reduced activity of tissue non-specific alkaline phosphatase, and also odontohypophosphatasia, which is a condition in which there is reduced activity of tissue non-specific alkaline phosphatases, and this is the rare autosomal recessive disease in which there is a marked deficiency in the presence of both acellular and cellular cementum. And here you can see the periodontal disease, which is very common in humans, and the most important clinical condition affecting the periodontal ligament is chronic inflammatory periodontal disease, in which toxic products are released by dental plaque lying in a gingival crevice and interacting with the host defense mechanism, including gingival clavicular fluid. These products result in the destruction and the loss. Here you can see destruction and the loss of periodontal ligament tissue and adjacent alveolar bone, and such a process leads to the formation of periodontal pockets, allowing for the accumulation of more plaques, and a vicious circle is established, and the loss of attachment tissue may expose the root in the mouth, and with increasing mobility, the tooth may eventually be lost. And here is one more, it is Epstein-Barr virus, which affects over 90% of humans and is usually transmitted by oral secretion and blood. And the virus replicates in epithelial cells and in B cells of the oropharynx. And most common symptoms of infectious mononucleosis are fever, lymphadenopathy, pharyngitis, or oral ulcers, palatal petechia, and less commonly gingival ulcerations. And that is all. And yes, have you any questions? Mhm. Let's then. This is today we discuss the periodontium and the two parts of periodontium. You know that periodontium consists of the cementum, periodontal ligament, and alveolar bone, and the gingiva. We discuss periodontal ligament, root formation, and cementogenesis. And for next lecture, we will deal with the development of orofacial and oral cavity, and at the same time the alveolar processes and alveolar bone. And the future next lecture is the last lecture of this course, and I sent you about 100 questions for exam. I make such a questionnaire for exam and I'll send you. I upload, and if you prefer, I send you. Any questions about this exam or questionnaire? Mhm. Then see you. Bye.